talk digestion. So we're going to ignore the mechanical part of digestion. We're strictly going to look at the chemical part of digestion. We break up the chemical part of digestion based off of which macromolecule we're actually talking about: carbohydrates, lipids, or proteins. Chemical digestion of carbohydrates. Look at in terms of chemical digestion is lipids. Lipids digest via lipases. We have two principal sites for lipase production. One is in the oral opening, salivary lipase. Salivary lipase is principally going to go after the small chain fatty acids. All the other large fatty acids that we absorb, that we digest, are going to be interacting with bile secretions. In that, we interact with the bile salts. Remember, bile salts is simply going to be an emulsifying agent. It's going to allow for the hydrophilic and the hydrophobics to interact with each other. It does that because it has two distinct regions. It has a hydrophilic region and it has a hydrophobic region that allows us to produce a vessel for the lipase to interact with the lipids. If we don't get this vessel here, we're unable to have the lipids fully digest. So when we do the lipase reaction, one of the lipase reactions is going to be done without bile salts, and one's going to be done with bile salts. And so we should be able to have the hypothesis that the ones with the bile salts will end up having more digestion than the ones without bile salts. The enzymes of importance here: lingual lipase, small fatty acids. Bile salt stimulated lipase needs what? Bile salt stimulated lipase needs bile salt. Pancreatic lipase doesn't come from the pancreas. Hepatic lipase doesn't come from the liver. Gastric lipase doesn't come from the stomach. The gastric lipase doesn't really digest much, but what it does is it curdles. So it takes the kind of liquidy lipids and it causes it to clump together almost like cottage cheese in terms of the coalescence. And then we have another couple guys, the lipoproteases, the PLPs. So how's it end up working? So we ingest lipids. They're going to interact with lingual and gastric lipase. They're either going to clump together, coalesce, coagulate, or they're going to get broken down into their triglycerides, diglycerides, or their free fatty acids. Very rarely are we actually going to get all the way down into here. They're going to pretty much stay as large fatty acids. They interact with bile salts, and we get the formation of an emulsification ring. So here is the emulsification ring. Here is the circulation of the fatty acids. Here is the rest of the bile salt, stabilized by lipoproteases and cholesterol molecules. They get digested down by pancreatic lipase, hepatic lipase, and bile salt sensitive lipase into either short chain fatty acids or referred to as long chain fatty acids. These long chain fatty acids tend to coalesce into a vesicle around themselves to protect themselves from the watery environment. That is referred to as a mycele. The mycele, when it gets absorbed and turns into what's referred to as a chylomicron. So here is the mycele. The mycele is stabilized by cholesterol molecules and proteins. And what it ends up doing is it ends up basically creating a hydrophilic region on the outside and a hydrophobic region on the inside. Same thing happens with the cholesterol molecules floating around the blood.